Fellow beekeepers, good evening, and especially warm welcome to this evening's speaker, Marla Spivak, who's one of the world's leading researchers from the Bee Lab at the University of Minnesota, where she's, I think, sitting now 5 hours away from us. So it's really good afternoon, Marla, to you. This is one of the many webinars that BIBBA have organised to help educate beekeepers. There are a lot more on the website and the recordings, and a lot more to come as well. I'd like to mention the next 2 weeks, which are really for people who want to move up from the amateur, out and out amateur, to the sort of semi-commercial. So if you are in that category, or you know anybody, then please please tell them. Also, just mention that Live at the Hive, which is me handling my own colonies at the bottom of my garden, they will be starting soon. So if you haven't registered, please register, and then you'll be advised when the sessions are. For this evening's presentation, please put questions in the chat function, but questions only, please. Not a load of chat, because we've got to sift them out. Marla this evening is going to be speaking about social immunity in honeybees, how colonies keep themselves healthy. So I'll hand over to Marla, and I'm sure you're in for a very good time. Marla, welcome.
Thank you very much, Roger, and thank you for inviting me, and hello to everybody in BIBBA and beyond. So yeah, I'm sitting in my office at the University of Minnesota. It's there's a lot of snow outside. It's March in Minnesota, which means it's both winter and spring at the same time. And I'm going to share my screen now. Let me get organized here. And Roger, if you could just tell me if you can see the screen. I think so.
Yep, that's fine.
Okay, great. So I've been giving this talk, or a version of this talk, quite a bit lately. So I decided to do something else for this group, and you can tell me in the chat afterwards if this works for you or not. I'm going to zoom way out and talk to you a little bit about ants and honeybees, but I'm going to use ants to frame the talk, and you're going to wonder why ants. We're beekeepers, we're bee breeders, but I hope this works for you. So I'll just begin. Social immunity, this term, was coined in 2007 by Sylvia Kremer. She's in Austria. She's an excellent researcher. She's, I think she's brilliant, and she does study ants. She coined this term. It means the behaviors, the organizational strategies, and physiological mechanisms that social insects use to defend against parasites to maintain the health of their colony. So this is used by ants, by termites, and by our honeybees. The question is near and dear to us as we're going through COVID, hopefully through COVID. But when you live in a densely populated society, especially an insect society where the colony can be 10, 50,000 individuals or more, how do you prevent disease transmission? And that is what the study of social immunity is. And here she is, Sylvia Kremer, my hero. And she published this paper in 2009, actually with her husband, who is a medical physician. And they were talking about the evolution, or the analogies in the evolution of individual and social immunity. And so they explain that, individuals, like this little mouse here on the left, has its own individual immune system. And the way you fight off disease actually begins with step number 1, prevent entry, which is why we wear masks, or have been wearing masks. And then number 2 is, if you get an infection, you fight it with your immune system if you're an individual. And if you're a social insect, you fight off that infection both with your immune system and by the behaviors of individuals. And then the third line of defense is trying to prevent that that pathogen from getting into the germline or your eggs and sperm. And in that, for bees or wasps and ants, that would be into the queens and males. And so her point is that in addition to the individual immune system of bees, in our case, when a colony gets together, their behaviors of individual bees are analogous to cells within the immune system, in that they're able to detect a foreign microbe and they're able to destroy it or encapsulate it and break it down. So the degree of social immunity changes as the disease progresses. So there's different stages, as Sylvia Kramer would explain. Bees, you have this amazing, and ants, but have this amazing division of labor where just by their age and the tasks they're performing, they limit their interactions with all of the nest mates. So that's just through division of labor, they can prevent disease transmission somewhat. But then they move into these other behaviors. She calls them prevention and sanitary care. That's like a preventative healthcare system. And then once it's infected, what do you do? What do these individual bees do? So some preventative care and sanitary care may involve disinfection, nest hygiene, grooming. And then once it's infected, you'll see some sacrifice of infected members. Okay, so let me give you this one example in ants, which was just amazing. And this was all done in her laboratory. These are little ants, Lasius neglectus. You probably have them there. And they live in the ground, ants do. And so they're exposed to soil pathogens all the time, including Metarhizium, which is a fungal pathogen that lives in the soil. And so their method of preventative care, if one of their pupae, shown here on the right, this diagram here, gets some of this Metarhizium on them, the ants in the colony start grooming the pupal cocoon, okay? So they groom it off first. And then they do a second thing, which is quite remarkable. They start spraying venom on the silk cocoon of the pupa to disinfect it. Venom is highly antimicrobial. And so they can kill off some of the Metarhizium right from the pupal cocoon just by spraying resin on their little babies. However, if the Metarhizium, the fungus, penetrates the pupa, then the ants start doing the second step, which was sacrifice. And they'll start, what they do is take that pupa and completely remove the cocoon. And then they start dismembering the pupa completely. They'll pull it apart and they inject venom directly into the pupa. And then they put it in a portion of their nest where they, a refuse pile where they put all their sick and dead. They don't throw it outside because other ants might bring it right back in. Honeybees, if they do any sacrifice, you'll see how they do that soon. They just throw it out the front door because once it's on the ground, other bees wouldn't bring it back in. And interestingly, when they're doing this unpacking of the pupa, when they're removing that cocoon, they're doing this when the pathogen, the Metarhizium fungus, is in its non-contagious form, when there's just mycelia growing through the pupae. And so they're able to unpack, dismember, and inject the venom into the pupa to prevent disease transmission. And they're also not transmitting because it's not infectious at that time. And so I'm going to be, the rest of this, I promise you I'm done talking about ants, but I'm going to use that framework to talk about preventative care and sacrifice in honeybee colonies and the two forms of social immunity they use. So their honeybee preventative care, I'm going to argue, would be propolis collection, and sacrifice, once they're infected, would be hygienic behavior, or the removal of sick, dead, Varroa parasitized brood. So I'll begin by talking about propolis. So propolis really is resin, and resin comes from plants. So some plants, not all plants, secrete these resins, and it's a defense for the plant. These resins are highly, highly antimicrobial. They're antibacterial, antifungal, antiviral, and they protect the plant, the growing plant tissue, from infection They also protect against herbivory and UV light. And honeybees are able to cue in to these plants that produce these resins and that are different than flowers. They're not going to flowers, at least in temperate regions. They're not going to flowers for resins. They're going mainly to the leaf buds, the growing leaf buds of certain plants. They scrape the resin right off of that plant and they pack it into their hind legs. As you can see in this left-hand photo, this is a bee in Brazil. It's scraping a green propolis off of this green plant, which is Baccharis dracunculifolia, very famous green propolis. And then they carry that resin load back to the nest. So the second photo with this bee with yellow propolis is a bee in our area that collected some yellow resin. And I'm using resin and propolis synonymously, and you'll understand why in a minute. Then they take the resin load, and it's so sticky, they can't get it off of their hind legs on their own. So a nestmate yanks this resin load off with her mandibles, and then together they cement the resin into the nest cavity, where then we call it propolis. So actually, resin is actually exactly the same as propolis. Bees are not changing the chemical composition of resin. They sometimes add a little bit of the salivary gland secretion. They can add beeswax to it, but the antimicrobial compositions are not changed once it's in the colony. So we knew about propolis, humans knew about propolis long before they understood it came from plant resin. So it ended up with two different names. These resins and propolis are full of terpenes, phenols, flavonoids, very complicated chemistries. So these are just beautiful pictures that someone sent me of bees collecting a red propolis and then a bee helping remove it from the other's leg. Tom Seeley pointed out years ago in his book from 1985 that in where bees nest naturally in tree cavities, they line the whole interior of the tree all around their combs with a layer of propolis that he called the propolis envelope. So on your left is a bee tree that the colony died. Here's was the entrance, and we opened it up, and this is the remains of the beeswax combs, and everything below this red dotted line, this brownish coating, is the propolis envelope surrounding all of the combs. In our beekeeping equipment, and we're using Langstroth equipment, and it's planed very smooth pine wood inside, bees do not create a propolis envelope. Unlike this tree cavity on the left, which is that same tree, and you're looking like as if you were a bee looking out the front door, and the combs are removed again, but this is that very thick, this orange, brown, green, yellow, very thick layer, almost half inch thick, is that propolis envelope, which you do not see in beekeeping equipment. We see bees putting propolis under the frame rests, between the boxes, in cracks and crevices, everywhere where it gets in our way. But so we've had almost probably in the United States over 100 years of selection against bees that collect a lot of propolis. It is a genetic trait, it's heritable. As far as bee breeding, you can definitely and easily select for high propolis collecting colonies. And I hope to show you that that would be a great thing to select for in your breeding program. A little bit about the biology. It was so interesting because this resin collection is extremely hard for bees. It's very sticky. They're not getting a food reward when they're collecting it. So why collect it? If they need to just seal up cracks and crevices, they could use wax, right? They produce wax. And yet they still, even in the United States with selection against propolis collection, they still gum up the works in our bee boxes. So they must want it and need it for a reason. And this photo just shows you the difference between a pollen load and a resin load so that you can look for these resin loads in your own bees. The pollen is fuzzy and very round, kind of symmetrical formation on the legs, and the resin is very shiny and it can come in different colors. I'm just showing a yellow load, but kind of not symmetrical at all. And the resin kind of catches your eye as you sit at the entrance and look for them. They're not very common, these resin foragers. Sorry. So over the years, knowing that it's so hard for the bees to collect, these students shown here at the bottom have been asking these various questions. And in particular, Mike Simon Finstrom was the first student in my lab to study propolis, and he continues to study at his position now as a researcher at a USDA bee research lab in Baton Rouge, Louisiana. And so you might be interested in the work that he continues to do. But in general, these students ask these questions: what are the plant sources of resin in our area? Where are the bees collecting the resins here? And what's the benefit of having that propolis envelope? Does it help them reduce disease load? And because it is so antimicrobial, how does the propolis affect beneficial bacteria and beneficial microbes that bees have in and on them as part of their microbiome? So just to begin with the first one, this is Mike Wilson's work that he collaborated with some plant chemists here at the university. He found that bees in our area are collecting almost all of the, resin from the genus Propolis, and mostly from eastern cottonwood, Propolis deltoides, also a little bit from balsam poplar and hybrid poplar that we have in our area. And what they're doing is going to these leaf buds where the leaves are just budding out, and they're scraping the resin right off of the leaves. And he figured this out using LCMS, liquid chromatography, followed by mass spec. And he collected resin from the plants and also resin loads from individual bees, and then matched them up chemically through these chromatograms that you're looking at on the right, and using all kinds of fancy statistics to see if they really matched to determine what the source of resin was. We have a lot of conifers in our area, pine and spruce, but we did not see any bees collecting resins from any evergreen conifer tree, just these deciduous cottonwood trees. In your area, it may be horse chestnut and other trees, but I'm not really sure. Sorry, it's okay. So to answer another question of after what happens, does it help the bees reduce the disease load? So what we did was create colonies with a propolis envelope. And in this case, what Mike Simon Finstrom did was collect propolis and make an extract. He dissolved the propolis in 70% laboratory grade ethanol, made a 15% solution, and just painted it in these little boxes here, these little 5-frame nucleus boxes. So this one has that green propolis. It looks brown here, but the green propolis from Brazil. And this is a local Minnesota propolis from cottonwood trees. And this box is just painted with the ethanol extract as a control. And then we would put bees in the boxes, and then we challenge them with chalk brood disease, Ascosphaera apis, a fungal pathogen of the brood. And we did that by taking these chalk brood mummies and homogenizing them and putting them into a pollen patty, feeding it to the bees. And within 2 weeks, colonies develop signs of chalk brood within the combs. And then we just sit and count the number of chalk brood mummies in the combs in colonies that have this propolis envelope or have no envelope. And this is on average 15 versus 108 mummies. It was quite remarkable and statistically different. Somehow the statistics got left off this page, but I think you can see without them. So it didn't cure the disease. It didn't eliminate the chalk brood completely, but it really helped reduce the disease load. Another way we made a propolis envelope was by using commercial propolis traps. This is these plastic grids that usually you put on top of a colony to collect some propolis within these 7-millimeter gaps. And in this case, this is Renata Borba's work. She cut up the traps to fit the inside of the box, all 4 sides of the bee boxes, stapled them in there, and then allowed bees to put propolis in the cracks over the whole season. If you remove the traps, this is what the propolis, quote, envelope looked like. For our experiments, we left the plastic traps in for the whole season to allow the bees to continue to bring in propolis through those gaps. I don't suggest you do this in your colonies. These traps are a little pricey and it really interferes with the bee space. So These are experimental methods. So I was just trying to show you how we created a propolis envelope for our experiments within our Langstroth boxes. So the results, but then the next thing she did was to challenge these colonies with American foul brood. So this is, of course, is a bacterial pathogen of the brood caused by Penibacillus larvae. And she sprayed a sucrose solution with Penibacillus larvae spores on each of the combs and presented them to colonies that had this propolis envelope created by the propolis traps or no propolis envelope. And to see what happened after they all developed signs of American foul brood. And you can see again, the maroon colored bars are colonies that had the propolis envelope, and the gray is colonies that had no propolis envelope. And this is a severity score, a 1 being less than 5 cells containing American foul brood per frame, and the 3 is well over 20 per frame, so very severe infection. You can see they all got sick with American foul brood, but the ones with the propolis envelope, the severity was quite lower, significantly lower compared to colonies with no propolis envelope. This is quite remarkable because they're only getting the volatiles and maybe a little bit of contact, and it's actually helping the brood within the cells. In general, propolis helps the bee's immune response, and we've done quite a few experiments on this, but I'm just showing you Renata's data where she created those propolis envelopes using the traps. We would collect 7-day-old bees from these colonies with and without a propolis envelope, and then look at their individual immune gene transcripts for immune-related genes. The names are hymenoptacin, abasin, defensin, etc. And the response from bees with a propolis envelope is shown again in this maroon color. And you can see compared to bees with no propolis envelope, the immune response was always lower. So whether the graph goes up or down, it's always lower than the gray bars, significantly lower in each situation. And over the season, that lower response was less variable. So bees in colonies with no propolis envelope, we saw their immune response go up and down and up and down. But with the propolis envelope, not only was it lower, but it was more consistent. And the reason is that having this propolis envelope is the propolis, the antimicrobial properties of the propolis is doing some of the job for the bees. So they don't have to activate their own immune systems because the propolis is killing off the opportunistic and pathogenic microbes, disease-causing microbes that are in the nest cavity. So the propolis envelope is like this first line of immune defense, killing off all these opportunistic pathogenic microbes in the nest cavity. And then if you additionally make the bees sick, then their own individual immune systems kick in. And together with this external immune or antimicrobial envelope and the bees' own immune systems, the disease load is very much lowered. So this is a good thing for bees to have a quieter immune system. But it led us to wonder, what about beneficial microbes? What about the microbiome? So this is a diagram of a bee with the crop and the ventriculus and the intestine, and in blue, the rectum. And of course, each section of the gut has its characteristic microbiome community, a collection, in this case, of bacteria. We're just learning to know the functions. But we looked at these bees, again, that were in a colony with a propolis envelope or without, and we sequenced all the bacteria that were in the bees. And what we found, okay, sorry, that's not there. What we found was every time we did this, that the abundance of the beneficial bacteria in the gut was higher compared to bees from colonies without a propolis envelope. So having the propolis envelope was actually helping the beneficial bacteria to grow. We have another study, and I'll show you the data from this one. In this case, we only looked at the microbiome from the honeybee mouth parts. And if you think about it, we chose the mouth parts because honeybees get their mouths into everything. They feed larvae with their mouth parts. They're collecting nectar with their proboscis, their tongue, and they're feeding each other mouth to mouth, sharing food, trophallaxis. They're tending the queen. So Holly, a master's student in collaboration with Kirk Anderson here, a researcher from a USDA lab in Tucson, Arizona, Holly collected bees from colonies with and without a propolis envelope, froze the bees, and then clipped off the mandibles and the proboscis. And then we extracted the DNA, and then Kirk Anderson sequenced all the bacteria that we could find on the mouth parts. So I should say the microbiome is not only in the guts, it's also all over the bee's body, just as it is in our guts and all over our skin and in our mouths. So we have microbiome species all over us. We're mostly microbiome, actually, as are the bees. And what they found, this is the data. This over here, this relative OTU abundance shows the percent of the bacteria. The bacteria in green are the core beneficial microbiome species found on the mouth parts. And the ones in blue are either pathogenic or opportunistic, and some of them are listed down here. And you can see bees from colonies with a propolis envelope, P+, had many more of the core microbiome species and significantly fewer percent of these pathogenic and opportunistic bacteria compared to bees from colonies without a propolis envelope. Again, indicating that having propolis helps the immune system and the disease load and helps the beneficial microbes in the guts and on their mouth parts amplify and stabilize. We did not find that the propolis envelope lowered the levels of Varroa mites, unfortunately, nosema or viruses. However, other researchers have seen a little indication that it does lower Varroa in the lab, but I'm going to show you some new data coming up shortly. In South America, they've found when they fed propolis in solution to bees, it would help reduce the nosema load. I would, if you're going to feed bees propolis, please know they do not consume propolis directly. So this is like feeding them a powerful antibiotic. So this would be used as a treatment for nosema. And also Drescher et al. From Germany found there was a little bit of a reduction in deformed wing virus in colonies that had a little bit of propolis. But very exciting research was just published this year out of Italy. And this, I don't know if she was a postdoc, Puchedu, in collaboration with Francesco Nazzi in that laboratory. What they found was so interesting to me, something I've wanted to know for a long time, and they figured it out, that bees are actually applying small amounts of resin on and maybe in wax cells. So this photo on the left is from that bee tree I showed you earlier. And this is the inside of the tree where the wood is. And you can see where they built the combs. You can see a little bit of propolis lining, resin or propolis lining on tops of the cells. But what we found is as the combs moved farther into the brood nest, we weren't seeing that propolis lining as much. But they found that even deep within the brood combs, and possibly that's why our brood combs turn this darker color with age, I really don't know. They found about 30 propolis compounds identified in the wax, some of the flavonoids, the phenols. And so they found some of those compounds, others were not found, and some were extremely concentrated within the wax. And so they did a very clever thing. They took gelatin capsules and they reared pupae within these gelatin capsules in an incubator in the laboratory. And in some of these gelatin capsules, they put a small amount of resin in there. and in others they did not. And then in ones with resin, they put Varroa mites or no. So they had this great experiment, and they found that in the gelatin capsules where they had both resin and Varroa on the pupae, it reduced the survivorship of the Varroa. So it increased the mortality by 20% and reduced the fertility of the Varroa by half. So this was quite remarkable. If this was really working against Varroa, none of our colonies would have Varroa, or they'd have fewer Varroa. But it does show that the propolis compounds are helping a little bit against Varroa, which is quite exciting. So they're getting a little bit of the resin or propolis into the wax, and it's also being infused into honey. So this is a quote from research by Maeve Birnbaum from a 2020 article. And it's a review article about honey, but she says, of the main phytochemicals or plant compounds that are reported in honey in northern latitudes, here are some of these plant compounds: pinobanxin, pinosembrin, quercetin, chrysin, galligan. Few of these are actually in nectar. You see them in honey, but not in nectar. And all of them are common in propolis. And even though propolis is not known to be ingested per se by the bees, ingesting honey may represent a major route of consumption by bees of these compounds. So honey may be getting infused to varying degrees by compounds from the propolis, as is the wax. And they're either directly applying resin to the wax, or possibly it's also getting infused through the volatiles. So it's very interesting. So the bees are consuming some of the compounds, but not the resins directly. So in general, a propolis envelope provides everyday benefits to bees' immune systems. It's very good for them. It helps promote their core microbiome species. It helps them reduce their disease load. And it evolved as a mechanical barrier, as a sticky mechanical barrier, and also in response to fungi and bacteria, these microbes within the tree cavity, we think. So right now we're just analyzing data from a big experiment, again, in collaboration with Mike Simone Finstrom, but also another graduate student, Maggie Shanahan, where we asked the question, how can we take these results and apply them to beekeepers? How can we stimulate our bees to create a natural propolis envelope instead of having to paint and extract or cut up propolis traps and staple them in and interfere with the bee space? So what we've done is collaborate with a guy who mills his own lumber, and he's made these boxes where the inside of each bee box is very rough. And this photo on the right, you can see there's 7mm grooves, just like the propolis traps, and it's a very rough cut. And when you put these boxes on bee colonies, we put bee colonies in the boxes, they fill these cracks and crevices with propolis, just like they do the propolis traps. So we gave some to a commercial beekeeper. We gave him 60 of these boxes. And we compared the colonies to 60 that were not in these rough cut boxes over the course of a couple of years. And these are preliminary data that we're just getting ready to publish. So these are the only data that are not published yet. And what we found by frames of bees, which gives an indication of colony population, the colonies that had those rough cut boxes that were propolis rich had slightly more frames of bees. So it's slightly larger populations compared to colonies without. But more importantly, I think, because beekeepers were asking me if we ask our bees to collect a lot of propolis, will it interfere with their honey production? And we found no difference in the amount of honey harvested. This is in pounds, and by this commercial beekeeper is quite a good harvest if the bees were in those rough boxes or not. And we did look at their immune system function and this third graph, and the more propolis they collected, the higher the propolis score, the lower their immune response, this abasin expression, which is what we saw in the laboratory, which again is a good thing. So that's a quick summary of all of our almost 12 or more years worth of data on propolis collection. And that I would consider, as I said, preventative care. And if you combine that trait with another genetic trait, hygienic behavior, I think as bee breeders, these two traits would really help bees stay healthy. And so using the ant terminology, I'm going to talk a little bit about hygienic behavior as a sacrifice of sick individuals. As you all probably know, this hygienic behavior was first studied in the 1930s and 40s here in the US by Park and Woodrow, and then again by Walter Rothenbuehler in the 60s when he looked at the genetics of the trait, by Martha Gillum and Steve Tabor in the 80s in relation to chalk brood. It is the ability of bees to detect and remove diseased brood. And again, just like the ants, they are detecting and removing both American foul brood infected and chalk brood infected pupae before the disease is infectious. So in the case of American foul brood, after the larvae consume the spores germinate and as they move through the gut. And in the form of chalk brood, the larvae are eating chalk brood spores, which germinate inside the bee into this vegetative mycelial stage. In both of those cases, during the vegetative or mycelial stages of bacterial or fungal pathogens growth, the bees are removing the pupae while they're not infectious. During those vegetative and mycelial stages are not infectious, only the spore stage is. So they're removing the source of infection before it's infectious, which limits disease transmission. I need to stress that, again, this critical detection and timing component of hygienic behavior, where they're able to detect and remove before it reaches the diseased infectious spore stage. We also know that hygienic behavior is one defense against Varroa. Again, the bees are detecting and removing the mite-infested brood, not just the mite, but the whole pupa that's infested. And again, there's a critical timing component to this because they remove after the mite has started laying eggs. So on the right, you see a foundress mite on the pupa. And if you look hard, you can see this is a male mite, if you can see my cursor, and some of the females down here and over there. So they uncap the cell containing mite-infested pupae and remove the whole pupa. The foundress may escape, but all of the mite progeny, as shown in this diagram, are killed during that removal process. So it reduces mite reproductive pattern and the mite reproductive growth in the colony. When we were breeding what we called the Minnesota hygienic line, we selected hygienic behavior based on this freeze-killed brood test. And I bring this up for a specific reason, but let me show you how this works. We would take these PVC pipes, we still do this, twist it down into a brood comb with sealed brood, and the 3-inch PVC covers about 160 cells of sealed brood or pupae. And we pour liquid nitrogen down the tube and let it freeze hard. And when the tube thaws, we put the frame back in the colony and check it in 24 hours. And what we're looking for is colonies that uncap and remove the freeze-killed brood within 24 hours. We always repeat that test to make sure we have no false positives. And then we would breed from colonies that are like that. We would raise colonies from these highly, highly hygienic colonies and not from those that still were removing brood at 24 hours. The rate of removal of that freeze-killed or dead brood is correlated with the rate of removal of diseased and mite-infested brood, but it's not a perfect correlation. You may be hearing now about Varroa sensitive hygiene, or VSH. And I wanted to just bring this up because the selection for VSH is quite different than selection using the freeze-killed brood. This is a line of bees. The VSH bees were first bred at the Baton Rouge USDA lab in Baton Rouge, Louisiana. And one way they used to select for these was to take a frame of brood from a colony that was highly infested with mites. And they would take this frame into the laboratory under a microscope and uncap 100 or 200 cells of brood and determine the percent infestation in the brood. Then they would take that frame and put it in a test colony and leave it there for a week, maybe 10 days, and then remove it and repeat step one. So they take that frame, same frame, back into the laboratory and open up an additional 100 or 200 cells and compare the percent mite infestation from start to finish. And you can see the photo of this colony, which is highly VSH, had uncapped many of the pupae that were infested with mites. And when their score of the percent infestation of the ones they experimentally removed was much, much lower. So what they're doing is actually looking for colonies that are able to detect and remove mite-infested brood, not just freeze-killed brood, but mite-infested brood. So it's very specific challenge and very difficult to do, not something that you and I want to do in our everyday breeding programs, I don't think. So where the research is going, and this is fascinating to me, based on this question, we began looking at many years ago, and others are also looking at quite in detail right now. How do the bees detect this disease and mite-infested brood? And we know that the bees are using their antennae and they're detecting odors associated with the pupae that are sick or mite-infested. And if they're highly hygienic or if they're highly VSH, from Varroa's sensitive hygiene, they can detect these odors at a very low stimulus level. So it doesn't take much odor coming from the brood for these bees to uncap that cell, poke their heads in there, figure out what's going on, and then remove it from the cell if need be. So researchers from all over the world are looking at these compounds that are emitted from mite-infested pupae. So these are researchers from France and New Zealand, and they found some specific cues associated with Varroa-infested brood. Another group from Canada found another set of compounds that trigger hygienic behavior coming from the brood. And then Olav Ruppel and Kara Wagner from North Carolina found yet a different collection of chemicals coming from Varroa and virus-infected brood. Quite fascinating. All of the labs, they used slightly different methods to collect and analyze these brood odors. All of these odors are probably correct. They're all probably associated with mite-infested brood. But the reason they're doing it, and this is work going to this last one by Kara Wagner and Olav Ruppel, they would like to find an easier assay for you, bee breeders, to use that's much more specific than the freeze-killed brood assay, and that would not involve you having to uncap and remove brood under a microscope and look at 200 cells in that very laborious way they select for VSH. So what we've been doing, and we did this a few weeks ago in US Northern California bee breeders colonies, we took some of those compounds that were identified, let me go back one slide, from this study, from Kara Wagner's study. And I'm skipping all over, I apologize about that. She had them synthesize these compounds, and then we put them in a little sprayer, and you can see it. I'm sorry, I don't have a very good spritzer. We just lightly put a PVC over 50 sealed pupae, kind of in the pink or purplized stage. And then we would spray 0.5 milliliter of this chemical blend on the cell cappings. We would spray it within the PVC, but I removed the PVC in this upper right photo, so you can actually see the spray going right on the cell cappings. And then we would mark that frame, put it in the colony, and come back at 2 hours, a very quick assay, and look at the percent cell cappings that were opened. And on the bottom right, you can see this colony had started to uncap well over almost 90% of the brood within 2 hours. So now we're looking at the results of, we're breeding queens from these colonies, and we'll be looking at them this summer at their mite loads compared to colonies that are selected using the freeze-killed brood test to continue Kara's work in a different university, different set of colonies. And so stay tuned. By the end of this summer, we'll see how well this serves as this little spray method, this unhealthy brood odor. We'll see how well it serves as a selection tool for you to breed bees that are more resistant to Varroa mites using this assay that's very specific to mite-infested brood. It's pretty exciting. Okay, so I'll finish up here just to summarize this concept of social immunity, which is basically how bees keep themselves healthy. They have many ways. They use division of labor and their interactions with nestmates to curtail disease transmission. They can prevent and they can sanitize their brood, and they can fight off infection once they're infected. And that's mostly through the hygienic behavior and removal of the diseased brood. So I think you'll all agree that bees are quite remarkable. Their healthcare system is quite sophisticated, and I'm in awe of them. I hope you are too. So I'll stop there. I want to thank my funding sources, of course, and the bees for collecting propolis and doing hygienic behavior, and the plants that produce the propolis, and all my wonderful students and technicians that make me look really good. I'm humbled by the work that they do with me in the laboratory. So I'll stop there. I don't know, Roger, if you want me to stop sharing my screen or keep it up, and I'd love to answer any questions you might have.
Thanks very much, Marla. That was fascinating. I think it's great these days that you researchers are actually sharing so much with beekeepers. I've been keeping bees nearly 60 years, and I think there's been a very definite shift in the attitude between researchers and beekeepers. Thanks very much. We have some questions come in.
I'm going to go ahead and stop sharing. Is that okay?
Yes. Yep.
Okay.
Yeah. Oh, I've got to find the chat wherever that is. The one that everyone is going to be asking is, we have always been told to torch and scrape our boxes out to get rid of all the pathogens and all the rest of it. It seems from what you're saying that it's probably best to leave the propolis there.
I think so. It is sanitizing the nest. If it's in your way, they're putting so much of it in certain places, if it's in your way, of course, you can remove that. But I think keeping it there is a good thing. I will say that over the winter, some of the antimicrobial properties are diminished. They're not lost completely, but it gets old. But as soon as the season starts again and the plants start growing, they'll collect more resin. So yes, I do suggest you leave it in there.
Do bees actually store propolis?
Do they store it? Well, yeah. I mean, they're not in cells, of course, but just within the nest box.
You do see great big lumps occasionally, don't you?
Yeah.
I wonder if that's what it is. I know bees collect from equipment that's left outside, like a queen excluder or something like that. So they do reuse it, don't they?
Yes, they do, and they'll rob it from each other also. They'll steal it from each other.
This one here, you can probably expand on it a little bit, but the question is, when in the year is the best temperature for bees to collect propolis?
Well, they'll collect it all through the growing season. As long as there's plants growing that produce the resins, they'll go collect them. I don't know if there's a minimum temperature. I think, as well as I do, what propolis is like in the colony. The warmer it gets, the stickier and runnier it can get. But, I think any foraging temperature, they'll start collecting it.
Another one which is interesting, as you said, the darker colour of comb could be attributed to the presence of propolis in the wax. Are we changing comb regularly for our aesthetic reassurance or for the benefit of our bees?
So are we replacing our combs? Was that the question?
Yeah, that's right. Yeah.
Well, I think we're replacing combs because there is a buildup of pesticides in there, and there's a buildup of pathogen spores in there. This is very new information that there might be any propolis in the wax itself. And I honestly do not know if the dark combs means there's a lot of resin or if it's just oxidation. I don't know why our brood combs turn so dark. I do think replacing combs is a very good practice. Bees in the wild don't use their combs forever and ever like we do as beekeepers. I think it's a very good practice to replace old combs periodically, every 3, maybe 5 years, maybe more frequently if you like to. Yeah.
I don't think you covered this, but there is a theory that bees actually coat the inside of cells with propolis. Is that correct or incorrect?
Well, that's what this research from Italy is saying. And I don't know if they coat the inside or just the rim, right? And I don't know if they're deliberately putting the propolis there or if it's just sticky footprints, tracks, their feet, dirt, quote, sticky feet on the on the rims of the cells. We really don't know this yet. So.
Does Marla have any recommendation of how to retrofit existing brood boxes to allow bees to create a propolis envelope?
Right. We've tried all kinds of things. We took a stiff wire brush and, roughen the surface, and that works a little bit. They'll put a little bit of propolis in the rough surface. We've tacked up window screens and all kinds of wire, all kinds of things inside the boxes. And eventually with window screens, they do fill it in with a little bit of resin. But, I think you beekeepers are much more inventive than I am, I think. And I think if you know that the inside of boxes should be roughened up, and the rougher, the better. The deeper the groove, the more propolis they'll bring in. I think you'll come up with your own ways. But do remember that, think of those propolis traps with that 7 millimeter gap and how much resin the bees put in those gaps. And I think that's what you're aiming toward. Think of the inside of a tree cavity, how craggy and how many crevices there are. And that's what the bees like. That's the kind of surface they want to put propolis on.
It's interesting you say that they don't put propolis on smooth surfaces, but I've removed ever so many colonies from wild places, and I've got several photographs of, so let's say, plywood buildings where the bees have actually propolized it. Yeah,
that is so interesting, and you're not the first person to tell me this. And so I wonder if it's your bees. So I know our US bees, most of them are not big propolis collectors, but I've seen bees from the Africanized bees and possibly your bees. Maybe they inherently collect a lot more propolis and are then therefore much more willing to put it on many other surfaces. Our bees collect so little that they're aiming for cracks and crevices, but maybe if they're really have the genetic tendency to collect a lot of propolis, maybe they'll go ahead and put it even on smoother surfaces. I don't know that plywood, is it as smooth as these pine boxes that we use? I don't know.
Just a quick question on the size of the groove. The 7 millimeter gap, is that the depth of the groove or the distance between the grooves?
Right. It's the width of the groove, 7 millimeter, it's the gap. But also I would go, I would go as, at least 7 millimeters, 5 to 7 millimeters deep.
Okay. How do you rate the 5-hour pin test method in the search for hygienic behavior?
How do I rate it? I think it's another way to select bees for hygienic behavior. And people have had very good success. I think if I were using the pin-killed brood test, I would come back within 6 hours to check the results and not wait 24 hours, simply because poking the hole does the first and hardest step for the bees. That detection and making that first little hole in the cell capping is the first step in hygienic behavior. And by doing the pin kill test, you're doing that step for them. So, I think it's, my ranking is for Varroa resistance would be this UBO odor, this new spray odor, using the actual compounds. The next most sensitive, I think, would be the freeze-killed brood, and just a little above the pin-killed brood method, where you're actually piercing and killing the brood. It's, of course, a lot easier, but I think what's easier for us is also easier for the bees. And I think we want to give them kind of a hard test.
This one's relative to human consumption of propolis, which I think you'll find a bit amusing. Is it okay for humans to eat propolis in its natural form, that is scraped off the hive and straight into the mouth? Well, when I've seen people do it, their teeth go brown.
I was going to say, it'll stick on your teeth. You'll be quite amusing looking if you eat propolis. But you can do it. And I think what you'll find is why the bees are not consuming resin directly either. I think it just is going to gum up their mouth parts where they wouldn't be able to get anything else in their mouths. It's so sticky. Usually for human consumption or medicinal use, it is dissolved in ethanol. There are some water extracts. I think the ethanol extracts are better at extracting some of the most of the compounds, the beneficial antimicrobial compounds.
This is an interesting one. We plant flora for nectar. Is it worth planting for propolis? And if so, what should we plant?
Yes, it is worth planting. In our area, we don't need to because we have so many cottonwood trees down by our rivers, populous. I guess you'd have to figure out what is the main propolis plant in your area. Is it chestnuts? Or I don't know what it would be. What other populus? You may have a different species of populus. And then figure out if you have a lot of them in your area. And if not, maybe grow them.
It's quite interesting because we used to have a matchmaking company in England. And about 60 years ago, they contracted farmers to plant poplar trees that they could cut down after about 40 years to turn into matches. And unfortunately, that company got bought out by a Swedish company. And there's a whole load of these poplars in fairly small areas, acre, 2 acres, something like that. And they definitely produce propolis.
That is interesting.
Is our intervention to treat with varroicides an interruption to the bee's natural hygienic behavior? I don't quite know what the question means there. But...
So if you're by treating bees with propolis, I think that was the first part of the question.
Yeah.
I don't know what
is our intervention to treat with varroicides an interruption of the bee's natural hygienic behavior?
Oh, I see what you're asking. For example, if you use a really stinky thymol or eucalyptol volatile, fumigant or whatever to treat for Varroa, does that interrupt hygienic behavior? I've never tested for hygienic behavior while the treatment is on, so I actually don't know. We always do our tests for hygienic behavior when there's no treatment on the colony. So whether formic acid or thymol blends would interfere, it may. However, if you think about it, the bees for hygienic behavior, they're the middle-aged bees, and they're walking over the comb surface and they're touching the cell capping with their antennae. And so the odor they're perceiving is slightly volatile or not very volatile, and it's coming, at a very close distance. They're touching it with the cell capping with their antennae. So having a bunch of thymol or formic acid in the nest cavity may not interfere with that level of detection.
Mm.
I guess you'd have to see for yourself, test it.
Okay. We know what mechanisms the bees have got for collecting nectar and pollen. You know, the recruit workers and that sort of thing. What happens with propolis? Do they do the same thing?
Yeah, can be the same workers that collect nectar and pollen, but on that day or that foraging trip, instead of getting food, they go out and collect resins. So then they'll, scrape the resins off the plant, bring it home, get it taken off their legs, and then cement it into the nest, into the walls of the nest somewhere, maybe on the rim of the cells. We don't really know. And then maybe on their next foraging trip, they might go out and collect pollen and nectar from different plants, completely different plants.
This is another interesting one. It's to do with patulines. Is it true to say that different groups of half-sister workers are responsible for identifying and uncapping problem brood and for removing the affected larvae?
I don't know if they're different patulines, but we do know that some bees are much more sensitive to the odors of diseased and mite parasitized brood than other bees. And they are the first ones to do the uncapping. And then the less sensitive bees do the removing. Here, I'll give you an analogy. If I walk through my kitchen and there's a cheese in the refrigerator that's going bad, I can smell it when the refrigerator door is closed. And I will open up the refrigerator and find the cheese, right? My husband, my dear husband, walk through the kitchen, would never know that there's anything going on wrong, and would be in the refrigerator with his head in the cheese drawer before he would say, oh, there might be something going bad here. So it's just a difference in olfactory sensitivity among bees, and whether they're different patulines or even the same patuline, just variation in olfactory sensitivity, we don't really know. It's good to have many patulines to ensure that you have bees that have tendencies to do all the traits that we need them to do. But I think even within a patuline, there's variation.
How do bees process or prepare resin to make propolis?
They don't. They just stick it in their boxes. They can add a little wax to it. And I understand they may add a little bit of salivary gland secretion. I don't know what it does, but they are not changing the resin at all. It's just resin in the nest.
Okay, well, I think that comes to the end of the question. So it's been a thoroughly interesting lecture. Thank you very much indeed, Marla. And we'll let you get on with your work now. So thank you very much indeed, and thank everybody else for watching.
Yes, and thank you very much. Let me know how you do with propolis. I'm very interested to know. Thank you.
Okay, thank you very much indeed. Good night, everybody.